Hi, I'm Dr. Wong. Sorry I can't meet with you in person to present these lectures. The digestive system is something that's really interesting for me because it is a complex system that brings in a lot of the other physiological systems that you learned about. Things like hormones and nerves from PSL 301 play a really important role in processing and moving material through the GI tract. You also have interactions between the digestive system with the cardiovascular system, with the renal system. So it's a really good way to end up the course because it can tie together a lot of these different systems. So whenever you talk about the digestive system, you're probably used to thinking of that long tube, that mouth, pharynx, the esophagus, the intestines, that modified tube, that's the stomach. But in addition to this, you've got really important accessory structures, things like the teeth, tongue, salivary glands that help to process that food, make it into a bolus, into a ball that can then move into that tube. And things like the liver, pancreas, those really important accessory organs, things like gallbladder that stores bile that's going to be used for processing of that food once it reaches the small intestine. And so there are a lot of different components to that GI tract, to that digestive system, that ensure that it can function properly. And we're going to be learning about these systems as we move through. And so that initial entrance into that GI tract is through the process of deglutition or swallowing. And so deglutition is just a fancy word for swallowing. And there are actually two phases to deglutition. There's a voluntary phase and then an involuntary phase. And so that voluntary phase starts whenever that food goes into your mouth and the teeth and the tongue move it around and you break up the food into a bolus or a ball of food. Deglutition is initiated whenever you make the decision to swallow that food. And so you make a conscious decision and you push your tongue up against the hard palate. Whenever you do that, that bolus, that ball of food gets pushed into the back of your mouth where the soft palate is. And it's that bolus of food pushing up into that soft palate that's going to be read out, that's going to trigger those sensory nerves like cranial nerve 9 and 10, the glossopharyngeal and the vagus nerve. That's going to take that sensory information up into the swallowing center and then activate nerves that are going to come down and influence things like the tongue, things like the pharynx, muscles around the pharynx, muscles in the esophagus. Now what's interesting is the upper 1/3 of the esophagus is made up of skeletal or skeletal muscle. And so you've got somatic nerves controlling that upper 1/3 of the esophageal skeletal muscles. The lower 2/3 of the esophagus are made up of smooth muscle cells. And so you've got the ANS, or the autonomic nervous system, actually acting on most of that esophagus. So once that bolus of food, you've pushed the tongue up against your hard palate, that bolus of food gets pushed into the soft palate, that food now enters into the esophagus. And as it passes into the esophagus, you'll have one quick last inhalation, and that has to last until the next stage. And so respiration is inhibited once that bolus of food gets deposited into that pharynx via a reflex. The involuntary phase of deglutition or swallowing occurs once that material is in that pharynx. And again, that sensory nerve, the vagus nerve, is carrying that information up into the swallowing center. And a number of things are happening. So things like the soft palate are going to be pushed up and close off the nasopharynx. You also have muscles that are going to contract and pull the larynx upwards. that movement of that larynx upwards is going to help to close off the trachea. So that food that's going to be passing along isn't going to enter into the trachea. You also have sphincters in the esophagus that are going to allow for that food to transition. And so that upper esophageal sphincter is going to open, allow that bolus of food to enter into the esophagus. Now, as that movement of bolus happens, the upward movement of that larynx through muscle activity, as well as that bolus of food pushing down, are going to help to close that epiglottis together so that food doesn't move into the trachea. It will stay in that esophagus. And so the major activities that happen with involuntary swallowing is closing of that epiglottis, but also peristalsis. And so we're going to be talking about peristalsis in a lot of detail in the next lecture, and we'll also talk about that esophagus, upper and lower esophageal sphincter that helps to isolate that tube as that food passes through it. But let's keep talking about those tubular structures, and the stomach is just a very specialized tube. It's specialized due to the fact that it's expanded. And that's because the stomach just stores that ingested food and allows for processing of the material that's been deposited into it. Now, it's very, very specialized because it has 3 muscle layers. There's a circular muscle layer that is going to allow for contraction of muscles and a change in the diameter of the stomach in one direction. You also have longitudinal muscle layers that are going to go the length of the stomach, and so you might see that contraction of a longitudinal muscle is going to shorten that stomach. And then you also have a partial layer of muscle called the oblique muscle, and that kind of runs diagonally. It's incomplete, but that now allows for contraction of that stomach to close down the diameter by the circular muscle, shrink the length using those longitudinal muscles, and another direction is via those oblique muscles. And so this way you have a really strong churning activity by these muscles, because part of the process of the stomach is to mechanically break down that food that you've delivered through your esophagus. There are also various cells, we'll talk about in more detail a little later, that are secreting things. They're secreting things into the lumen or into the inside of that stomach, and that is also going to help to chemically break down that food. Transition out of that stomach is going to be controlled by a sphincter. The pyloric sphincter controls movement of material only when it's ready out into that small intestine. Now, movement of material from the esophagus has been controlled by another sphincter here, but this is called the lower esophageal sphincter, and we'll talk about that in more detail. But this portion of that tubular GI tract, the stomach, is isolated by the presence of these 2 sphincters that coordinate flow of material into it and out of it. Whenever we get into the small intestine, we'll be talking in a lot more detail about some of these cells, but you've got a lot of cells lining that small intestine in a layer called the mucosa or the mucosal layer, and we'll talk about that in the next slide. But I wanted to tell you about these very, very specialized cells that populate the small intestine. And so there are epithelial cells. They can be called absorptive cells. Sometimes they're just called enterocytes. These are going to do a lot of different things. Some of them are there just to facilitate movement from the inside of the gut across the cell to then be deposited across that basolateral surface into the body. There are some of these cells that secrete things like bicarbonate as well. And there are other structures that are or material that is being secreted that we'll talk about a little later. There are also a group of cells called enteroendocrine cells. These are just endocrine cells that are located in the small intestine. And so we'll learn more about those, and those are going to be secreting hormones. Remember, hormones are always secreted in the bloodstream. So enteroendocrine cells aren't secreting things into the lumen, into the inside of that GI tube. It's secreting material into the blood to carry messages around. You also have a number of different types of exocrine cells. Exocrine cells like goblet cells and Paneth cells. Those goblet cells are going to secrete mucus, and we'll see the role that mucus plays. Paneth cells are also secreting antimicrobial compounds. You might see the relevance of that in a minute. And then you also have a population of stem cells, and those stem cells are undifferentiated cells that can proliferate, and then they can differentiate into a wide range of cell types to replace cells lost. So the GI tract has a basic structure. There are 4 layers throughout that GI tract. Depending on where you are, it can physically look a little different. And so this is an area within the small intestine because of the way that they have finger-like projections called villi. We'll talk about those in more detail in this fold called a plica. But the general layout of that wall of the GI tract is the same to those 4 layers. So you've got a mucosa or mucosal layer that faces into the inside of that tube or into the lumen. So that mucosal layer, the mucosa, contains a layer of epithelial cells that form a barrier between the inside of the lumen and the inside of your body. So material has to be transported from your gut across that epithelial barrier to eventually be absorbed into blood and lymphatic vessels. There's a very loose layer of connective tissue called the lamina propria. In here, you've got a number of structures like mucosal glands that are secreting mucus. You also, in this area, have those finger-like projections we'll talk about in a minute and what they are doing and what structures look like, but I want to introduce you to the concept of these finger-like villi or finger-like projections called villi. So those are part of that small intestine. So that lamina propria is going to be underlying those villi, those finger-like projections, and that's just loose connective tissue within there. What's interesting is that there's also a very thin layer of muscle called the muscularis mucosae, and that contraction of that layer of muscle allows for these little finger-like projections to wave and move and change their position as they wave and allow for a bit of stirring of material in that lumen. The next layer is called the submucosa or submucosal layer, a much more dense layer of connective tissue. And what you have here is a lot of the blood vessels, lymphatic vessels that are going to be picking up material and also oxygenating material within that gut. Then you've got this really thick layer of muscle called the muscularis externa, and it is actually composed of 2 layers of muscle arranged in different orientations. So you've got a circular layer of muscle in that muscularis externa, and so if you think of a circular layer of muscle, it's going to contract and restrict the diameter of that lumen. You also have a longitudinal layer of muscle, and what that is then going to do is shorten the length of that GI tract in a localized way. And then associated with the muscularis externa is also going to be a nervous plexus, nerve system we'll talk about. And then finally, you've got a serosa, a serosal layer. The serosa is really just specialized epithelial cells that are secreting a number of different secretions that facilitate movement because it's like a fluid-like secretion, and so your intestines actually can move against each other, which is necessary because there's a lot of movement to your small intestines as they're processing food. And the serosa is really part of a larger mesentery, smooth mesentery that covers everything in your peritoneal cavity. Now, you also have a large immunological material. About 70% of the immunocytes in your body are associated with GALTs, gut-associated lymphoid tissue. So you actually have a lot of immune response ready to attack anything or ready to bring information about anything that you've ingested and the pathogens that might be carried among those. And so one of those GALTs is found in the lowest portion of the small intestine in these patches called Peyer's patches that are just collections of immune cells found in that region. And so you've learned about B cells, plasma cells, you've learned about macrophages, you've learned about CD4 T cells, and these are some of those cells that are found in those Peyer's patches. And just looking around in that lamina propria for anything that's entered from that outside of your body that you've ingested that's sitting in that lumen of your GI tract. Now, there are really interesting cells called M cells or microfold cells. And these M cells sample the lumen of your small intestine and pass information, pass material to immune cells found in the Peyer's patches, and then they can mount an immune response. These Peyer's patches are connected up to lymph nodes found within the mesentery that can also then transport information around. And so this is a great example of how blood and immunity, surprisingly, maybe not surprisingly, has a really important component in your digestive tract. Now, your gut, that GI tract, actually has its own nervous system called the ENS. The enteric nervous system actually contains as many interneurons as your spinal cord. And so it contains sensory neurons, it contains components of that autonomic nervous system, as well as interneurons that allow for the ENS to make decisions on what is happening in the gut and the response that that gut has to have. So the ENS really does integrate that sensory information through a bunch of neurons within that submucosal layer. You've got two branches. You've got the mesenteric plexus, and that mesenteric plexus regulates motility. It's also called the Auerbach's plexus. And it might make sense because the myenteric or Auerbach's plexus is found in between, layered in between that longitudinal muscle and that circular muscle. And so you can see how information is going to be relayed to the muscle via that Auerbach's or myenteric plexus. The submucosal plexus or Meissner's plexus is actually at that margin in that submucosal layer. And so it is going to regulate secretion and absorption. If you can think about where that's located as well, that's where all those glands are going to be secreted. It makes sense that it's going to regulate secretion. It's where material is going to come from the lumen, the inside of the GI tract. So here's the inside of the tube. And remember, material has to cross that epithelial barrier and get in and make its way in. And so a lot of that absorption information is also going to come from this submucosal plexus. So now start to think about how the digestive system is regulated. You're usually thinking that it's going to be via that CNS, via the cephalic brain, that is going to then send signals maybe down onto that myenteric, onto that submucosal plexus to cause things like secretion of material or motility patterns. But because I've just told you about the enteric nervous system, you might now start to understand that even local stimuli, things like distension of that GI tract, the presence of food being sensed, changes to osmolarity as material gets broken up. It'll change the osmolarity within the lumen of the small intestine and stomach, as well as the presence of acid that might be secreted from the stomach, and we'll be coming to that. But remember, all of those can be signals that are going to be sensed by those sensory receptors, flow through interneurons that are also going to have an effect on the myenteric and submucosal plexus to then cause things like motility patterns, release of pancreatic secretions, but also material being secreted from the stomach and small intestine that are then going to feed wider into the body, going to the brain, going to the endocrine pancreas to signal for hunger or satiation, or to signal we need more insulin and downregulate something like glucagon. And so the ENS allows for this local control where a signal can come in, a decision is made on it, and an effect is the response that you see. But you also do have this cephalic phase where that information, although it can stay in the ENS, can also be relayed up to the cephalic brain to then come back down into the gut to have motility and secretion patterns. And so you just have to think about that long reflex up to the CNS, to the brain, or a short reflex. Can it be handled totally within the ENS itself? So a great example of local control is endocrine secretions from the enteroendocrine cells within that small intestine. And so you have sweet taste receptors. These sweet taste receptors are formed by dimerization of T1R2 and T1R3 receptors. And so they will dimerize into these sweet taste receptors. And these are the exact same type of sweet taste receptors that you have in your mouth, but they're found in the small intestine. Now, what these sweet taste receptors do is signal, and they're going to signal with the binding of sugar, activate intracellular signaling in those enteroendocrine cells that they're attached to for the release of integrins. Incretins are peptides by definition. Incretins are hormones that are being secreted by the gut that are going to stimulate secretion of insulin from the pancreas. That's the formal definition of incretins. But these incretins, GIP, GLP-1, so glucagon-like peptide 1, glucose-dependent insulinotropic peptide, are both incretins. By definition, what they are doing is stimulating the release of insulin from the pancreas, but in this case, they can also act in a paracrine fashion. And so binding of sugars onto that dimerized sweet taste receptor found on enteroendocrine cells causes the release of GLP-1 GIP that acts in a paracrine fashion. And what they do is signal to the surrounding enterocytes to upregulate expression of SGLT1. You've already heard of SGLTs, sodium glucose linked transporters. And what SGLT1 on enterocytes do is allow for that glucose that stimulated the sweet taste receptors to move into the enterocyte to be absorbed. And so you've got this local control. You've got glucose activating the sweet taste receptor, signaling the release of GLP-1 and GIP. Part of what it does is act in a paracrine fashion and causes absorption of that sugar through upregulation of SGLT1. That sugar can now cross that enterocyte and be absorbed. You also have autonomic nervous system control of your GI tract. And so parasympathetic is rest and digest. And so generally that's going to increase that gut muscle activity, increase secretions into the gut to facilitate absorption, relax sphincters, relax those entrance and exit points through those various areas of that tube, so that material, that bolus, after it reaches the stomach, it gets broken up into something called chyme, CHYME, and that chyme then moves through and can be absorbed and eventually is going to get modified into waste. And so increasing secretion, increasing muscle activity to move that material through, relaxing those sphincters as they're required is going to help with that rest and digest response. You've actually got neurons, though, that are projecting to the gut, both from the brainstem, so you've got that vagus nerve coming down, as well as the sacral region of that spinal cord. And that is going to then impinge on various regions of your GI tract. So you can see that the efferent vagal nerve or fibers are going to synapse on neurons within the ENS. So they impinge right on that ENS in the esophagus, the stomach, and part of that small intestine, all of the small intestines, and part of the large intestine. Pelvic nerves coming out of the sacral spinal cord are going to impinge on the ENS in the lower area of that large intestine and within the rectum itself. And so these signals are going to be going directly to that ENS. What you have here is the release of acetylcholine and their muscarinic receptors that are going to be used to signal on those structures. You also have the sympathetic autonomic nervous system, and so this is going to be that fight or flight, and so innervation for that sympathetic is going to exit through the thoracic and through the lumbar regions of the spinal cord, and they are going to synapse in ganglia. So that means now you're talking about a preganglionic and postganglionic innervation. And so the preganglionic neurons are going to be releasing acetylcholine, and those are going to be releasing the acetylcholine onto the nicotinic receptors on these cell bodies in the ganglia. And so acetylcholine onto nicotinic receptors. The cell bodies within the ganglia are now going to form those postganglionic fibers, and those postganglionic fibers or neurons are now going to impinge on that ENS, and what that's going to do is release noradrenaline onto adrenergic receptors. And this relay of information is what is going to cause that fight or flight response, which means that you're going to shut down that GI tract, you're going to inhibit gut movements, you're going to reduce secretion, you're going to constrict those sphincters that would normally have allowed material to move from one region of the GI tract to the other. You also have a very specific portal system within your GI tract. So splanchnic circulation just describes the circulatory system or circulatory structures in your stomach, your intestines, your spleen, your liver. Splanchnic just means viscera or internal organs. But I want to introduce you to this concept of a portal system. And portal systems, by their definition, are just where you have one capillary bed connecting up to a second capillary bed in sequence. And so you can see that material is going to move through that aorta into one capillary bed. They're all going to come together again through the hepatic portal vein and then break up into another specialized capillary bed in the liver. And that's all going to join together into the hepatic vein that's going to go back into the vena cava, back to the heart. So that arterial blood supply is bringing that oxygenated material. So both the liver and that digestive system are receiving well oxygenated blood via that aorta. You've got a number of structures, and all I want you to remember is you've got the celiac trunk. It's going to be sending blood vessels to the stomach, to the spleen, but there's also a common hepatic artery. And that common hepatic artery is then going to divide up, continue to send blood to another region of the stomach. But what's interesting is this hepatic artery proper. And so the hepatic artery proper is going to be taking that well-oxygenated blood up into the capillary system within the liver. But you also have oxygenated blood moving through the mesenteric arteries coming off of that aorta that's going to go to the small intestine and to the large intestine. Okay, so just picture this as material going through to one of the capillary beds that we're setting up with our hepatic portal system. So this is material that's going into the stomach, oxygenated blood to the stomach, to the small intestine, and to the large intestine. Now, when you're at the small intestine, if you recall, I talked about villi, these finger-like projections that can be wiggled by the muscularis mucosae. So these villi are finger-like projections that have capillaries surrounding a specialized lymphatic vessel called a lacteal. And so here is one of the capillary beds created by that oxygenated blood coming in off of those various mesenteric arteries that we talked about, since we're talking about the small intestine here. So those capillaries are important for gas exchange, everything you've thought about from cardiovascular, but ultimately what is important for us in the digestive system is transport of the nutrients that have made their way across that epithelial barrier into either that blood circulatory system. Some of those structures have to move into the lymphatic system, and we'll talk about fat absorption in those central lacteals, or in those lacteal structures, a little later. But let's focus on the blood circulatory system. And so here, suddenly you've loaded these absorbed nutrients into these capillaries. And so now they have to make their way to the liver. So now we're going to start to bring material back, and it has to go to the liver to be processed. And so all of those mesenteric structures are going to mirror a venous and an arterial system. So we had the superior and inferior mesenteric veins going to the intestine, arteries, going to the intestines. Now we've got the superior and inferior mesenteric veins coming away from the intestine. You also have the blood vessels coming from the stomach. They all join up into the hepatic portal vein, and this is that connecting blood vessel going from the capillary bed in your GI tract, in the stomach, small intestine, large intestine, carrying that nutrient-rich material back to the liver. So that blood is going to be characterized by all of those nutrients that have been absorbed across that epithelial barrier that makes up that mucosal layer. So now we're going to take that nutrient-rich blood It's all going to combine from all of these areas into that hepatoportal vein and get to the liver. But remember, this is part of a portal system. What the capillary bed looks like in the liver are these sinusoids. So sinusoids are very, very specialized capillary spaces where oxygenated and deoxygenated blood are able to mix. They're also specialized because they have these open pores, so that material can easily leave those capillaries. And that's important because they are transferring material to hepatocytes, those liver cells that are then going to process that material. So blood comes in from your digestive tract through that hepatic portal vein. Don't forget, you also have the hepatic artery proper coming in, and those are going to open up into that capillary bed made up of those specialized capillaries we call sinusoids. As that blood travels along, it's going to exchange material with the hepatocytes, but eventually make its way into a central vein. Now, each of these functional units of the liver made up of all of these hepatocytes are called a lobule. And so a lobule has associated with it these hepatic veins and arteries at the specialized edges. Blood moves into that central vein in the middle of that lobule. And then from that central vein, all of that blood will collect into the hepatic vein and make its way back to the vena cava, back to the heart. Now, it has to have a 1-to-1 ratio or contact between hepatocytes and that sinusoid. And so this hepatocyte layout is extremely important to the functioning of the liver. Now, as material gets processed by that liver, some of the material gets dumped back out into the sinusoids, but some of it gets transported back towards the small intestine, contained in bile. And so you've got that bile ductules coming back from the hepatocytes, converging into bile ducts that is now going to move material back into your GI tract, whereas the blood is going to continue on back to your heart. So once we have that bile created, it is going to all combine into a common hepatic duct. That bile can drip down into the common bile duct and empty out into your small intestine. It enters into the small intestine through something called a sphincter of Oddi, and so that sphincter just has to be opened to allow for that bile to be deposited or dumped into the small intestine where it's going to aid in digestion. But you can also have that bile backing up into the cystic duct to be stored in the gallbladder. And that bile is going to be used in fat digestion, we'll learn about. In the gallbladder, any bile that has been backed up and stored in there is now going to be diverted and concentrated as water is resorbed from it. So we'll talk a little more about the pancreas and its role and entrance into the sphincter of Oddi whenever we talk about digestion. So here's what's happening in that liver. You've got material brought from that GI tract. So things like nutrients, any foreign substance, any ingested drugs, bilirubin that might be ingested as well is going to be brought back through that hepatic portal vein. Remember, you've also got the hepatic artery. So any kind of bilirubin that hasn't been ingested, but that's due to the breakdown of red blood cells, metabolites of hormones and drugs, any other nutrients that are floating around can also come back in through the hepatic artery to the liver. The liver, those hepatocytes are going to metabolize glucose and fat that has been absorbed from the GI tract. It's going to aid in allow for synthesis of proteins, of hormones, urea production, storage of material. You actually also have a number of cells and enzymes that are going to aid in detoxification. Depending on what has to happen to it, that material can go back out through those bile ducts, secreted into the small intestine. The earliest part of the small intestine is called the duodenum. And so we'll talk about what's in that bile, things like bile salts, things like water, things like phospholipids that are either going to be lost or utilized in digestion. You also have collected in those sinusoids, moving into that central vein, into the hepatic vein, to then gets pumped around your blood to peripheral tissues. Things like the glucose that you've absorbed, things like plasma proteins, you've learned about clotting factors, you've learned about things like angiotensinogen. You've got urea that has been produced as well, and that's going to be used by the kidneys. You've got vitamin D. If you took PSL 300, you know about vitamin D here. And then any metabolites for excretion are also going to make their way to the kidney. And so this is something that I'm not going to test you on, but I know that in the renal section you talked about at some level in the renal section these organic anion transporters, and you actually have a lot of those in your hepatocytes. And so things like antibiotics, therapeutic drugs are transported out of the bloodstream into the hepatocytes. And you've got things like your cytochrome P450 or CYP enzymes. You've got a number of types of dehydrogenases, other enzymes that are going to metabolize things like drugs, but also things like toxins. And those hepatocytes can detoxify and then eliminate those toxins through a number of transporting proteins, extrusion proteins that are going to dump them into the bile system to get dropped out into the feces. And so my point with this slide is you don't have to remember all of this information. You do have to remember that your hepatocytes are what is detoxifying you. You have evolved lots and lots of structures to facilitate transport of toxins into the hepatocytes. They detoxify, they get rid of the things. And so you do not need to eat things specifically to detoxify because your liver is doing that for you already. So that ends this lecture. Thanks so much for watching. If you have any questions, you can definitely email me. My email address is here. I'm also the coordinator for the course, so you can easily find my email address